Hello my dear students, how are you all? Hope all of you are fine. So in the last video I discussed the alternative forms of Mendel's dihybrid clause. Okay. So far in our discussion we have seen what was the work culture of the Mendel and what the work he has published. Okay. Now today I am going to start the next topic, that is chromosomal theory of inheritance. Chromosomal theory of inheritance. So what is the meaning of this one, that we will see, but actually we will make some more discussion about the Mendel's work. Okay, so you know that Mendel performed his studies on garden pea, okay, and he published his work in year 1864. He published his work in year 1864, but unfortunately his work was not recognized in year 1900. Means for 36 years his work was not recognized. You know what was the reason behind that? In the board examination this is very common question. What was the reason behind the unrecognition of the Mendel work immediately after he published the result? What was the reason behind that? Okay, so that you should know. So what was the reason? That three main reasons are there. First one is lack of communication. Lack of communication. Lack of communication and media, you can say. Actually nowadays you see this media and communication is well developed. Means whatever the work is done by any scientist, that is getting easily comes into the light because media and communication is now it is working smoothly and it is well developed. But during that time this method of communication was very poor. Means he was not having any proper channel to publish his result. So that was the first reason that is considered, lack of communication media. Second is concept of factor. Concept of factor was not accepted by contemporaries. Contemporary means the people, so those who were living that time. You know that instead of gene, Mendel used the term factor. But this concept of factor was not accepted by the contemporaries, means the scientists who were present at that time, they were not familiar with the concept of this factor, and moreover there was no physical proof for the presence of the factor. Then third reason that is considered for the unrecognition of the Mendel work is using mathematics in biology was a new approach. You know that Mendel used the concept of probability. Mendel used the concept of probability to solve the questions in the genetics, bio. And that was also a new thing. Means it was the first time that the concept of the mathematics was used in the bio to solve the problems. That was also not accepted by the contemporaries. Okay. One more reason you may find that is given in some of the books, that during almost at the same time Darwin also gave his theory of natural selection. Darwin also gave his theory of the natural selection, and that was also means the mass, or we can say the group of the scientists, they were busy in listening the theory that was given by the Darwin, survival of the fittest, or we can say theory of natural selection. That's why he did not get any attention from the contemporaries. So these three are the main reason, but that Darwin theory also have a role. Okay. So these are the main three, or we can say four reasons are there why Mendel work was not recognized. But what happened here in year 1900, I told you, his work came into light. So how his work came into light? His work came into light when three independent scientists, von Teschner, Hugo Davis, and Karl Correns. These three scientists, they independently rediscovered the Mendel's work. They independently rediscovered the Mendel's work. What happened? They also performed the hybridization studies and they also published the result. But later they come to know that the result that they are going to publish in the article, in the magazine, that similar result was already published by a person named Mendel. Then they were shocked that this result was published 36 years ago, and because of that they went in the search of the Mendel, but that time Mendel was no more. Okay, but they found the book of the Mendel in which he made all the studies. So from that they they come to the conclusion about the Mendel's work that we are studying now. Okay. Then what happened? By year 1902, you know that the concept of factor was also new for the biologists that time. So what happened? By the year 1902, chromosomal movement was also studied. Chromosomal movement was also studied, means how the separation of chromosome take place during the gamete formation, that you already studied in class 11th. Okay. How the movement of the chromosome take place during the gamete formation, that you already studied. But what happened? In 1902, the chromosomal movement was studied because of the development in the microscopy. So on that basis, this chromosomal theory was given, and it was given by Walter Sutton and Theodore Boveri. Name of scientist is very important. So the concept, this chromosomal theory of inheritance, this was given by Walter Sutton and Theodore Boveri. So Walter Sutton and Theodore Boveri, they gave chromosomal theory of inheritance. Actually, what they told in this theory is very important actually. They develop a parallel relationship in this chromosomal theory of inheritance. They develop parallel relationship between gene and chromosome. So they develop a parallel relationship between gene and chromosome. How they develop the parallel relationship, that we have to see. What they told, that whether it is gene or chromosome, both are present in the form of pair. If you remember in class 11th I told you that chromosomes are present in the form of pair, and that pairs are called as homologous chromosomes. Then on that homologous pair, that segments of DNA are there, and that segment of DNA, they are called as what? Gene. Here you can see the chromosomes are present in the form of pair. Similarly, genes which are present on the chromosomes, they are also present in the form of pairs. They are also present in the form of pairs. So what they told, they developed a parallel relationship between the gene and chromosome. They told that gene as well as chromosomes are present in the form of pair. Then what happened? During the gamete formation, whether it is gene or chromosome, one of the pair, only one of the member of the pair, because in the pair two are there, one member of the pair will be transferred to a gamete. When the means we can say when the gamete formation will take place, only one of the two will be transferred to a gamete, because you know when the gamete formation will take place, the gametes are haploid, therefore this chromosome will move in the one gamete, this chromosome will also move in the another gamete. So what happens when the gamete formation takes place, both of these two will separate, or we can say segregate, such that one of the two will move to the gamete. When these chromosomes will move to the gametes, what about these genes? Obviously, genes will also move towards the gamete. Clear? In NCERT, you can see on page number 82, you can see a comparison is given between the behavior of chromosomes and the gene. On page number 82 in the NCERT, you can see a comparison is given between the chromosomes and the gene. There you can see table first and second point is same. You can see it is given occurs in pairs in the A column also, in the B column also it is given occurs in pairs. Second point you can see in the A, segregate at the time of gamete formation such that only one of each pair is transferred to the gamete. Same point you can see it is given segregate at gamete formation and only one of each pair is transferred to a gamete. Same thing is given. Then third point you can see it is given independent pairs segregate independent of each other. If you remember, this is nothing but the concept of Mendel's law from the law of independent assortment, that independent pairs segregate independent of each other. In the B also you can see one pair segregate independent of another pair. And if you read downside it is given, can you tell which of these columns A or B represents the chromosomes and which of them represents the gene? So how will you come to know that which of the two represents the gene or chromosome? The difference you will find in the third point only. First and second point is same in both of them. Therefore difference you can find in the third one. Actually genes, the things about the gene is given in the A column, because you can see it is given plural word, independent pairs, because you know on a single chromosome many genes are there, so here the word will be used plural. But in the B column you can see one pair segregate, this one pair will segregate independent of the another pair, so that B column will be the chromosomes. So if suppose in the examination they are asking to explain the chromosomal theory of inheritance, simply you have to start from here only, okay? Don't start from the Mendel's work. If it is of 3 marks question, then you can start from the Mendel's work. But if it directly they are asking, then you may start from here only. That this theory was given by Walter Sutton and Theodore Bowry, and they develop a parallel relationship between the genes and chromosomes. Then you can make that table and show the relationship between both of them. Okay. Then the experimental verification, this is also very important. The experimental verification, because you know whatever the theory or hypothesis is given, until and unless the experimental verification is not done, it is not considered true. So even though both of them theoretically they develop the parallel relationship between the gene and chromosome, but the experimental verification of the chromosomal theory of inheritance was done by T.H. Morgan, Thomas Hunt Morgan. So the experimental verification of the chromosomal theory of inheritance was done by the T.H. Morgan. And he performed his studies on the very important Drosophila melanogaster. So he performed his study on the Drosophila. It is an insect. Then in the examinations, questions coming from the Drosophila also. Why Morgan or why Drosophila is considered for the work in the genetics? What is the reason behind that? So some 3 points I will tell you. First thing is that the Drosophila, their lifespan is very short, around 2 weeks. Means they complete their life cycle in 2 weeks. So what happened? In a small duration of time, multiple generation can be studied. In a small duration of time, multiple generation can be studied. Then second is they are easy to culture. They are easy to culture means they can be cultured on any synthetic medium. They can be cultured on any synthetic medium. That's why we are saying that they are easy to culture. Third point you should know that a single cross or we can say single mating produces large number of progenies. A single mating produces large number of progenies. So in that progenies you can study the inheritance of the trait. And fourth point is that the male Drosophila and female Drosophila can be easily distinguished. The male Drosophila can be easily distinguished from the female Drosophila. Okay. So what is the importance of this one? Usually the sex-linked character. The sex-linked character can be easily studied because until unless you are unable to identify the male and female Drosophila, the sex-linked characters cannot be studied. So these are the four reasons why Drosophila is considered as the experimental material in the genetics. Okay. So this completes the chromosomal theory of inheritance. Now from the next turn we will start linkage and recombination. Thank you.